I'm Dr. Harvey Jenkins, MD, PhD, retired orthopedic spine surgeon, pain management physician, and biochemist. Picture the blood vessels in your brain as a plumbing system, quietly carrying nutrients in and flushing waste out. In Alzheimer's disease, part of that plumbing rusts shut, and the waste that backs up is a toxic protein called amyloid beta, the stuff behind Alzheimer's plaques. A new study out of Monash University looked at a copper-based compound called CuATSM. Rather than attacking plaques directly, it repairs the pumps that clear amyloid out of the brain in the first place, pumps called P-glycoprotein. In mice with Alzheimer's pathology, CuATSM increased those pumps by 24%, cut toxic amyloid beta by 42%, and improved memory performance by nearly 44%. If you followed Alzheimer's news, you've probably heard of lecanemab and donanemab. Those are antibody drugs, and they work like a demolition crew, latching onto plaques that have already formed and hauling them away. CuATSM does something different. Instead of clearing existing plaque, it tries to fix the leaking pipe so the brain can clear amyloid on its own, continuously. Those approaches aren't necessarily competitors. You could imagine a future where one clears the existing backlog while the other keeps it from coming back. Researchers also suspect CuATSM may rev up microglia, the brain's resident immune cells, which act like an internal cleanup crew that engulfs and breaks down plaque debris. If that holds up, CuATSM could be attacking amyloid from two directions at once: better drainage out of the brain and a more active cleanup crew inside it. Here's what makes this compound interesting to me. It's not new. CuATSM has already been studied for years in two other neurodegenerative diseases, and that history matters. Let me explain a protein you'll hear me mention twice: SOD1, short for superoxide dismutase 1. Under normal conditions, SOD1 is a protective enzyme. Its job is to neutralize free radicals, unstable, damaging molecules that are a natural byproduct of cellular energy production. Think of free radicals as sparks thrown off by a cell's machinery, and SOD1 is the fire blanket that's supposed to smother them before they cause damage. In about 2% of ALS cases, mutations cause SOD1 to fold into the wrong shape, like a key bent slightly out of true. Misfolded copies clump together, and those clumps are toxic to motor neurons, the nerve cells that control muscle movement. CuATSM delivers extra copper directly to these motor neurons, which helps misfolded SOD1 refold closer to its correct, harmless shape, acting essentially as a molecular chaperone. In animal studies, this slowed disease progression and extended survival, in some models outperforming riluzole, one of the few FDA-approved ALS drugs. Researchers even found disrupted copper levels in spinal cord tissue from sporadic ALS cases. Patients without the SOD1 mutation DAOs, suggesting this approach could help a broader population. CuATSM has since advanced to human phase 2 and 3 ALS trials. In Parkinson's disease, dopamine-producing neurons die off in a region called the substantia nigra. Researchers studying post-mortem brain tissue found something unexpected. Copper levels in that region were significantly lower than normal, along with reduced activity of copper-dependent enzymes, including SOD1 itself. That flips the usual story about metals in the brain. Here, the problem was a shortage, not an excess. CuATSM showed neuroprotective effects across 4 animal models of Parkinson's by topping off that copper supply, and it progressed to a small human phase 1 trial in 2019. The thread connecting ALS, Parkinson's, and now Alzheimer's is oxidative stress, cells struggling under chemical damage, often tied to failing mitochondria. CuATSM appears to specifically target that distress, wherever it shows up in the brain. To be clear, the Alzheimer's data is from mice, not people, and no human Alzheimer's trial has been announced yet. Earlier research also found that higher doses of CuATSM cause side effects in some human and animal studies, so dosing will matter a great deal going forward. But because this compound already has a safety record in humans from ALS and Parkinson's trials, it may have a head start most new drug candidates don't. I'll be watching to see where this goes. Thanks for watching. For more scientific breakthroughs, please like and subscribe and visit us at unbedsidemanners.com.